Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my lecture. So today we're going to discuss some more important questions from this chapter, molecular basis of inheritance. So this is a chapter you have highest weightage in your class 12. So in class 11 it is your human physiology. So I thought I will take one more section to discuss the important questions. So I have almost 20 questions that we will discuss. So it's going to be a very short session. So thank you so much for watching my session today. So please don't forget to like, share and subscribe. Come on, quickly do share the link with your friends. So let your friends know here we are discussing all important chapters, all important questions. Come on, make it quick. Yes. So molecular basis of inheritance, so we have taken ample time to finish this chapter in detail. So we spoke in detail about everything, the process of replication, the process of transcription, the process of translation. So we spoke about everything in detail. So now we want to discuss questions, and yes, it is a tendency that the questions come tricky and you as students will make it a bit wrong. So you might go wrong in the silly mistake. So yes, good evening Pushpa, can I score 340 plus by listening your class? So you're asking 340 plus in biology? Yes, Pushpa, thank you for joining my lecture. So please do like, share and subscribe. So if you're asking me if you want to score 340 plus in biology, my classes alone are not enough, so your efforts are also equally important. So I want you to read NCERT textbook every day, followed by my PPTs. So my explanation might be just a help. So please read NCERT. Once you listen to my lecture, then please solve questions, and only then you will be able to score marks. So only my classes is not sufficient, because my class is just explanation, right? So how you understand it matters. And yes, I want you to read NCERT as well. So please go through your NCERT. Yes, Shamita, hello, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Alright, so let's go forward and see what I have in store for you. Okay, so today once again we are meeting at 6 p.m. So here I'm going to talk about the effective strategies to prepare for NEET exam. Like your NEET exam is nearing, so you people are getting anxious, you people are getting tensed, right? And everyone has this biggest doubt, how to start the preparation, how to kickstart the preparation, what is the most effective way, how should I revise, what is going to happen, what should be the mindset of a student, so how should you go about for the next 2 months? So all that we are going to discuss in this interactive session. So please join me at 6 p.m. this evening. So do share with all those who are appearing for the NEET exam, so it might help them. Alright. Yes, Monica. Oh, hello. Very happy good evening. Anthony Abhi. Yes, good evening. Thank you guys for joining my lecture. Yes, Anthony, I'm good. Thank you. How are you? So please do like, share and subscribe. Okay, so please make sure you share this link with your friends and join my session at 6 p.m. So yes, so once again for all the aspirants who are appearing for NEET exam 2021, so the crash course is kick-starting from tomorrow, that is 15th of July. So this will happen in eCareerPoint app. So this is completely in English medium. So physics, chemistry and biology will be taught in English medium. So biology I will be handling. The offer price is still be the same because your NEET exam is nearing, so they haven't changed it. So it is 1,999. So every day you have 1 hour lecture of each subject, followed by your doubt discussions on Saturday, and Sunday you have your unit test. So please make sure you join us for the same. The course is commencing tomorrow. Alright? Okay. So the topics that we discussed in this chapter were your central dogma of life, your replication, your translation, the regulation of gene expression, what is DNA fingerprinting, then we spoke about your human genome project, right? So these are some of the topics that we discussed in this chapter. So any doubt, any particular part you want me to teach once again? Ma'am, in NEET there is an internal choice, please explain in the evening class. Yes, the NEET paper has a bit modification, which also we will address in the evening class. Yes, Anthony Abhi, done. Okay. Yes, any particular topic that you want me to explain in this chapter? So we started off talking from the structure of DNA. So we spoke about the structure of your DNA. So then we spoke about experiments, for the search of genetic material. So it started from your transforming principle. Then we spoke about the central dogma of life. So this includes replication, transcription, and translation. So in translation, we spoke about your genetic code as well, right? So then we went on to talk about your gene expression, regulation of gene expression. Yes, then we went on to talk about your human genome project, and then finally about DNA fingerprinting. So these are all the topics that we discussed in this chapter. So do you want me to recap any particular topic? Yes, please ask me now. This chapter is equally important. Yes, Dharni, a very happy good evening. Thank you for joining my lecture. So please do like, share and subscribe. Yes, any doubts? So in replication, we spoke about Michelson and Stahl's experiment. Yes, Naveen, hello, hi, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So which means you're all quiet, so everyone is clear in everything. Okay, so Anthony Abhi wants me to explain wobble hypothesis. So what is wobble hypothesis? So you have your mRNA, right, and you have your tRNA. So this is the anticodon arm in your tRNA, right? Yes, Shampi, hello, hi, a very happy good evening. Yes, I'm good, thank you. So please do like, share and subscribe. Okay, so this is your anticodon arm of tRNA. Fine, and this is your mRNA. So this is 5' to 3'. Suppose if I have a base AUG here, okay? So this is the first base, second base, third base on your mRNA, but it is the other way round on your tRNA. So what is the complementary sequence here? The complementary sequence here is UAC, right? So in your tRNA, this is the first base, the second base and the third base. Right, so first it base pairs here, right? So this determines the specificity. So this is your wobbling hypothesis. So mostly what happens, your last base pair changes. So that, which means, for example, if you have 4 codons which codes for the same amino acid. For example, let's take UG as common. Okay, UG is your stop codon. We take UC, UCG, UCC. So let's say all this codes for the same amino acid alanine. So what changes is your last base pair, right? So this is prone to changing. This is the same, right? Only this base pair changes. So this is wobbling. So wobbling depends on the base of third codon on your mRNA and the first codon on the tRNA. So this is wobble hypothesis. Anthony, did you understand? What is wobble hypothesis? Yes? Others, any other topic that you want me to explain once again? So please ask me. Yes, come on. Anthony, you're done? You understood? Shall we go to the questions quickly? Yes, is it clear? Okay, so let's go to the questions that we have. So quick, so we are already done with the first 10 questions. So I am going from question number 11. So please start solving. Okay, Anthony Abhi, thank you. So what will be the sequence of mRNA produced by the following stretch of DNA? So this is my DNA. So they have given my template strand, my coding strand. So what will be my mRNA? Yes, what will be your answer? Make it quick. Question number 11. How do you find it within 2 seconds? There is a trick. What is the trick? Yes, what is the trick here that you should follow? So, this is the strand from which we are going to copy the information onto the mRNA. Right? So, your mRNA will be complementary to this. So, this is also complementary. So, your mRNA will be similar to this, except that wherever there is T, you replace it with U. The answer is the same. Right? So, your mRNA will be similar to your coding strand, except that the T will be replaced by U. No. Why? Why? We are copying this strand. How will it again be AUG? No, so you have 2 strands like this, right? So we ideally write 5 prime to 3 prime on the top. So this is my coding strand. So this is my template strand. So I copy the information from this strand. So these 2 are complementary. Yes? These 2 are also complementary. So your mRNA and this coding strand will be the same, with the only difference that T will be replaced with U. So option B is the right answer. Okay? So please don't make mistake. So question number 12. So coding will be this. We will take only the template strand. So template strand is where we are copying the information onto the mRNA. Okay? Yes? Is that fine? Okay, so question number 12. So in bacteria, which enzyme catalyzes the transcription of all type of RNA, that is mRNA, tRNA as well as rRNA? DNA dependent RNA polymerase, DNA dependent DNA polymerase, RNA dependent RNA polymerase, RNA dependent DNA polymerase. Yes, question number 12. Question number 12. Catalyze the transcription. So transcription is when you copy information from your DNA to RNA. So this is transcription, right? So transcription is done by RNA polymerase, no doubt. But the template here is DNA. So it is DNA dependent RNA polymerase. So when do we say this dependent? It depends on the template. For transcription, the template is DNA. So the right answer is DNA dependent RNA polymerase. Okay? So question number 13. What happens in the tailing process of transcription? Adenylate residues are added at the 5' end of RNA. Adenylate residues are added at the 3' end of RNA. Guanylate residues added at the 5' end of RNA, or guanylate residues are added at the 3' end of RNA. Yes? Yes? So tailing is a process that happens after your transcription process. So you have capping, you have splicing, and then you have tailing. So capping is at the 5' end by guanosine methyl triphosphate. So splicing is where your introns are spliced off. And tailing is at the 3' end where you add adenylate residues. So now what is the answer? Yes, absolutely right. So it is at the 3' end. Shimpi, it is at the 3' end, not 5' end. Yes, Swami, a very, very happy good evening. Thank you for joining my lecture. So please do like, share and subscribe. So question number 14. Fully processed hnRNA to undergo translation is called as what? So fully processed hnRNA which undergoes translation is called as. So hnRNA is known as heteronuclear RNA. So when it is fully processed, meaning it is processed with capping, splicing and tailing. So this is the complete process. It is known as your mRNA, right? So that is what we call as your mature mRNA. So the right answer is option A here. Okay, everyone agrees? Can I move forward? Yes? Okay, so this is the answer for question number 4. tRNA, so how did tRNA come here, Shimpi? So Shimpi is trying all different options A, B, C, D. Hannah, Shimpi? Yes? Okay, so here we go, question number 15. So yes, hnRNA is the precursor of mRNA. You're absolutely right, Anthony. So choose the correct option. So splicing represents the dominance of the RNA world. The presence of introns is the reminiscent of antiquity, the remain of the ancient. Split gene arrangements represent an ancient feature of the genome. All of the above. So which statement here is right? Quick. Which statement here is right? Yes? Yes, ma'am. A very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Yes, Shimpi, all of the above is right. So your favorite answer, right? All of the above. Okay, so perfect. So this is question number 15. So question 16. So if there are 999 bases in an RNA that codes for a protein with 333 amino acids and the base at positions 901 is deleted such that the length of the RNA becomes 998 bases, how many codons will be altered? So they are saying you have 999 base pairs. Okay, so divided by 3 because our codon is triplet, right? So totally we will have 333 amino acids, right? So now they are saying if the base at 901 position is deleted, then what will be the final outcome? How many amino acids will be changed? So if one amino acid changes, the entire amino acid after that the reading frame changes, right? Sorry, if one base changes, then after that the reading frame changes. So the remaining 99 base pairs will be affected. So 99 divided by 3, so it will be 33 codons which will be altered. So they are altering in terms of, sorry, they are asking in terms of codon, so it is 33. So the answer is C, not D, it is 33. Anthony Abhi, did you understand? Arun Balaji, yes, hello. So thank you for joining my lecture. Please do a video and KVPY in biology with important topics. Okay, you want me to do talk about KVPY? Okay, suggestion taken. So I'll think about it. Thank you. Yes, so here the right answer is option C. I hope you understood this calculation. Yes? Okay. So they are saying there are 999 bases in your RNA. Right? So 999 bases. So I divide by 3 because my codon is a triplet. Right? So which means totally I should get 333 amino acids. Now they are saying the base pair in the 901 position is deleted. Okay, so it is like this, the hat is red, the red hat for suppose. So if the 901, suppose this is the first position, if this is deleted, then how it will be? So everything changes after this, right? So if the 901 base pair is deleted, all the remaining 99 base pair will be changed. The reading frame will change. So thus how many codons will be altered? 33 codons will be altered. So after this I have 99 bases, right? If 101 is deleted, the remaining was according to me 99. Right, so 99 divided by 3 because my codon is a triplet. So they are asking in terms of codons. So 33 codons will be altered. Yes, did you understand, Anthony? Hi Munna, yes, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. Yes, Anthony, you understood? Yes, okay. So question number 16. So removal of RNA polymerase 3 from nucleoplasm will affect the synthesis of. So we have three types of RNA polymerase. So RNA pol 1, RNA pol 2 and RNA pol 3. So if I remove 3, whose function will be affected? So RNA pol 1 is used in the synthesis of rRNA. So pol 2 in the synthesis of hnRNA, which is a precursor for mRNA, and this is for your tRNA. Yes, what is your answer? Yes, right, okay, fine. So question number 18, UTRs present on mRNA refer to untranscribed regions at both 5' and the 3' end, untranslated regions at only the 5' end, untranslated region at both the 5' and the 3' end, untranslated region at the 3' end. Okay, so both these options are same, so it should be translated regions. Yes. What is the right answer? Okay, so it's right only, I'm sorry. Yes, it's not same. Yes, what is the answer? Okay, so Munna and Shimpi, wow, how beautifully you both are talking, huh? Okay, yes, the right answer is option 3 because this is transcribed and this is translated. UTR is untranscribed regions. Yes, absolutely right. So perfect. So question number 19. So which enzyme will be produced in a cell in which there is a nonsense mutation in the LAC-Y gene? So in your lac operon, we have 3 structural genes, right? So Z, Y and A. So this codes for beta galactosidase. So this codes for permease. And this codes for transacetylase. Yes, so if you have a nonsense mutation, a mutation that has no function, if it's nonsense mutation in the LAC-Y gene. Okay, so what is this type of mutation? What is this type of mutation? Nonsense. It does not make any sense. So if you have a mutation in the Y gene, yes. So if it's a nonsense mutation, the process of transcription will end. So then only beta galactosidase will be produced. So this will not be produced and your RNA polymerase will not go to transacetylase. So the right answer is option A, beta galactosidase. So if there is a mutation in the Y gene, right, it is a nonsense mutation. So RNA polymerase will get terminated here. So only beta galactosidase will be produced. Alright, yes? Fine. So please get it right. So why it is option A? Everyone got it right? Yes? Any doubt? Should I explain once again? Done? Anthony Abhi, did you understand? Swami? Yes, is it clear? Okay, Shimpi, fine. So question number 20, percentage of similarity between nucleotides of 2 individuals is. So what is the similarity between Shimpi and Munna? So 2 individuals who are a bit closely related, but still they are different. So how close they both are? Questions are like fire. Wow, thank you. Yes. Yes. So percentage of similarity between the nucleotides of 2 individuals are, so we spoke about this in your DNA fingerprinting. Yes, quick. Yes, absolutely right. So it is 99.9%. We are all similar. So the 0.1 is where we all differ. So that is why I am different. That is why you are different. Yes, absolutely right. So question number 21. So SNPs can be used for finding chromosome locations for disease-associated sequences, tracing human history, evolution, all of the above. Yes, question number 21. So SNPs are single nucleotide polymorphisms. Okay, it is all of these. They can be used to trace human history as well, because always we all differ only in the SNP, and if there is a difference then it leads to evolution. So all of the above is the answer. Yes, Shimpi, your favorite answer is right here. Okay, yes, fine. So question number 22, VNTR varies in size from. So VNTR is your variable number of tandem repeats. So what will be the size? This I have not discussed, but what do you think will be the size? So it is repeats, right? Suppose for example, if this is the repeat, so what will be the size? This is very short, but something like this. It is repeated continuously. So it will be from 0.1 to 20 kilo base pairs. So please make a note of it. The size of VNTR will be 0.1 to 20 kilo base pairs. Okay? Yes? Yes, NCRT line. Yes, perfect. So question number 23. The bulk of DNA other than repetitive forms the major peak during density gradient centrifugation. So the other small peaks are referred to as, so we have the bulk DNA, so that is your genomic DNA. So this is without repetitive DNA. So it forms a major peak like this. So the other small peaks form what? Other small peaks form what? Satellite DNA, non-satellite DNA, exonic DNA, intronic DNA. Yes, question number 23. So the other, so this is without repeats, so the other one should be with repeats, right? So it will not be like this, it is satellite, so it is around. So the major peak that you have is your genomic DNA. So the other small peaks or the other peaks that you have will be your satellite DNA. So the answer is option A. So satellite DNA refers to your repetitive DNA. I am sorry. Okay? Yes? Fine. So question number 24, the basis of DNA fingerprinting is high degree of polymorphism in sequences, low degree of polymorphism in sequences, intermediate degree of polymorphism in sequences, sequences show no polymorphism. Yes, question number 24. Yes, Shubham sir, hello, hi, very happy good evening. So thank you so much for joining our lecture today. So please do like, share and subscribe. Yes, what is the answer for question number 24? What is the basis of DNA fingerprinting? So I told you the basis of DNA fingerprinting is single nucleotide polymorphisms. Right? So single, yes, absolutely right. So single nucleotide polymorphisms, they show high degree. So high degree of polymorphism in sequences. So that is the basis of your DNA fingerprinting. So this is your SNPs. Alright? Yes? Okay. So moving to the last question. So VNTR belongs to the class of satellite DNA referred to as microsatellite, mini satellite, mega satellite or repetitive DNA. Yes, what is the question? No, I still have some more questions. Yes. Yes. So VNTR is a type of satellite DNA. So satellite DNA is of two types, right? One is mini satellite and one is micro satellite. So VNTR belongs to your mini satellite. So the right answer is option B, okay? Yes. So please make a note of this if you haven't understood. Can I move forward? Yes, clear? Yes. So question 26. In Escherichia coli, lac-operon is induced by lactose, promoter gene, beta-galactosidase I gene. Lac-operon is induced or activated by. Question number 26. Yes, quick. So who acts as the inducer in your lac-operon? So what induces the transcription of the three genes ZYA? So here the inducer is lactose or allolactose, right? So the right answer is lactose. No. So promoter will induce your, okay. So promoter gene will induce your the promoter gene will go forward to your inducer and it produces a repressor mRNA, so which in turn produces a repressor protein, right? But this repressor protein will go and bind to the operator. But to induce your lac-operon, that is to induce the transcription of this, you need a lactose because lactose holds this repressor, right? So it is your lactose. No, it is your lactose which acts as an inducer here. When there is no lactose, lac-operon is not functional. Only when lactose is there, it is functional, right? Okay, so question number 27. Lac-operon is arabinose operon, is a repressible operon, it is an inducible operon or it is an overlapping gene. So what is a lac-operon? So just now we spoke in the previous question. So your answer should come in handy. Yes, quick. Lac-operon is arabinose operon. Lac-operon is lactose operon. Shimpi. Yes, what is the answer here? Quick. What is the answer? Come on, quick. Lac-operon is inducible, right? Only when there is lactose, it is getting turned on. It is an inducible operon. When lactose is there, its concentration increases. So the right answer is option C. Yes, so question number 28, yes, do the matching quickly. So match the column 1 with column 2 and select the correct combination. So Griffith gave the transforming experiment, right? Prokaryotic DNA is your nucleoid. So Hershey-Chase experiment proved that DNA is the genetic material. So they used a bacteriophage. So this is transduction. Then your euchromatin is the active chromatin. So what is the right answer? A4, B3, right, C1 and D2. So the right answer is option C. Yes, perfect. Yes, good. So make it quick. So don't delay in answering. If you can do the question quickly, do it quickly and then cross check. Okay, done? Yes, question number 29. So mutations which alter nucleotide sequence within a gene are called as frame shift mutation. Heterochromatin is the one which is inactive. Euchromatin is transcriptionally active. Hetero, it is closed. U is open for transcription. So mutations which alter your nucleotide sequence within a gene are called as frame shift mutation, base pair substitution, both A and B or none of this. Question number 29. Yes, heterochromatin stains dark. Yes, you're right. What is the answer here? So what can be the possible answer? Quick, question number 29. Come on, guys. So question number 29. Yes, it is called both the frame shift mutation as well as base pair substitution. Yes, absolutely right. So here we go to the last question for the evening, question 13. So each individual has a unique DNA fingerprint as individuals differ in the number of mini satellites on chromosome, the location of mini satellites on chromosome, the size of mini satellites on chromosome, and all of the above. So why do we all differ in DNA fingerprints? What is this most probable reason? Yes, quick. Quick. Yes, Shimpi, right. Your all of the above today seems to work well. Yes. Good. So the right answer here is all of the above. It is because the number of repeats that I have is different and the location of my repeats is different from yours, and the size of my repeat and the size of your repeats are also different. So with this we are done with the molecular basis of inheritance completely. So from tomorrow you have a change in timing. So tomorrow at 4:30 we are playing a quiz. It is from the second unit of your class 11, that is morphology, anatomy and structural organization. Okay, so it is season 2 and episode 2. So we have a Menti quiz for 25 minutes. So tomorrow, that is the only class we have, and from tomorrow is Thursday. So from Friday, so our classes will be from 5 to 6 as of now. If there is any change, I will let you know, because now I have reduced the number of classes because you should also do the self-study. Right? So any one day I'll bring 2 classes when you need a motivation session. Yes, tomorrow you have a Menti and day after tomorrow you'll have a lecture. So we'll start a new chapter. We will start excretion. So we are yet to start with excretion, right? So we are done till body fluids. So there will be a change of class. So the number of class will be reduced because I want you to do self-study. And if you want some revision classes, if you want some one-shot series, so please write to me in the comment section, alright, so that I will know what is your need and accordingly we will plan. So I will do the testing or the revision series from 1st of August. Yes, we can finish it off in 30 days. We'll only do the important chapters. So if you have any suggestions as to which topic you want me to teach, so please write to me in the comment section, not in the chat box. Okay? Yes? Yes, you will have only 1 session per day. So any one day I will take 2 sessions because it takes up a lot of your time to come here, sit, read from me. You have a lot of distractions as well, right? So if you have self-study, that gives you a lot of relaxed mode so that you can concentrate on your own. So in a day we will have 1 session, that is revision, so all the time you can revise. Yes? Is that fine? Okay, so please do write in your suggestions like what kind of classes you want. So whether you want one-shot series, whether you want me to teach any topic in detail. So please send in me a suggestion and give me 2 days so that I will plan up and get back to you. Okay, so please think about the crash course. So it is worth it, and please join me for my class at 6 p.m. this evening, right? So if you genuinely liked my way of teaching, so please do like, share and subscribe. If you only want me to revise only the previous year question or the most important question, so the ones that we discuss now are critical thinking questions. If you just want me to discuss the most important and the most repeated questions, please type in the comment box. Alright, so thank you so much for making my session very, very interesting. Yes, thank you Shimpi, thank you Anthony. So take care and I'll see you at 6 p.m. So until then, bye.